When grown in suspension, neural progenitor cells isolated from neurogenic niches of the brain proliferate to form neurospheres. Here we show the isolation and serial sectioning of free-floating postnatal hippocampal-derived neurospheres. To locate protein expression within the three-dimensional neurosphere, we will perform immunostaining for a protein of interest, in this case connexin 29, followed by assembly and translation of two-dimensional microscopy images into a three-dimensional reconstruction of the neurosphere. Visualization of proteins within the context of their native three-dimensional structures, such as a free-floating neurosphere or a tissue, allows for a better understanding of protein function based upon location within these structures, and also allows the inference of possible roles for proteins with known localization but unknown functions. Hi, I'm Sophie Imbeau from the Neural Regeneration Laboratory at the University of Ottawa.
And I'm Nicholas Valenzuela from the Carleton Immersive Media Studio at Carleton University in Ottawa.
Today we'll be demonstrating how to isolate, expand, and serially section hippocampal-derived postnatal neural progenitor cells as neurospheres in order to localize protein expression via immunostaining.
We will then use two-dimensional images of the serial neurosphere sections collected following immunostaining to illustrate a method of reconstructing cellular and protein location in the three-dimensional structure, and thereby assess spatial organization of protein expression throughout the neurosphere. So let's get started.
Once brains have been collected, prepare to place them onto the chuck by creating a flat surface for the brains to properly rest. This is called blocking. Block the cerebellum using a razor blade and forceps. Next, put a drop of crazy glue onto the chuck and fix the first brain onto it, anterior side up and dorsal side facing you. Proceed like so with the other brains. For the last brain, it is prudent to position the dorsal side away from you, as cutting is sometimes incomplete in this direction. If incomplete cutting should occur, use a scalpel to slice through and collect the section. Speed is of the essence for the gluing step, as the brains can't be exposed to air for too long. Once all brains are glued in, the chuck is placed in the vibratome and the holder filled with ice-cold ACSF. Set the vibratome for a frequency of 8.5 and a speed of 3.5 to 4.5. Bring the blade into contact with the ACSF and set the automatic cut. Now we will cut 500 micron slices. Keep the slices which contain the hippocampus. Place them in a dish filled with ice-cold ACSF and discard the rest of the slices. Once all slices are collected, proceed to the hippocampal dissection using a stereo microscope. We only take the dorsal hippocampus approximately between bregma minus 1.65 millimeters and minus 2.10 millimeters. Place the hippocampi in an empty ice-cold dish. When all the hippocampi have been collected from the slices, it's time to bring them into the flow hood for mechanical dissociation using a scalpel. Spray the scalpel blade with 70% ethanol and flame to sterilize. Cut the tissue with the scalpel until it appears liquidy. Now, using dissociation media already containing papain, DNase, and neural protease, collect the tissue by pipetting and place into a 15 milliliter tube for enzymatic dissection. Place the tube in a rotating oven set to 37 degrees and let digestion take place for 45 minutes to an hour. Once digestion is complete, add 10 milliliters of sterile tissue culture PBS to stop the enzymatic reaction and spin the cells down at 300 times G for 5 minutes. Remove the supernatant and resuspend the pellet in 5 milliliters of PBS. Use a pasture pipette and triturate the sample to further dissociate the tissue. Keep triturating until the pieces of tissue no longer get smaller. At this point, pellet the cells again. Remove the PBS and resuspend the pellet in 3 milliliters of maintenance medium. Once you have well triturated the sample, take a 15 microliter aliquot and mix it with an equal volume of trypan blue. Take two 10 microliter samples and load a hemocytometer for cell counting. Count the cells from 3 fields in each section for a total of 6 fields. Average numbers and calculate your cell density, keeping in mind the sample is diluted in trypan blue. Plate the cells in low adherence dishes at a density of 2.5 times 10 to the 5 cells per dish in a final volume of 5 milliliters of maintenance medium. If low adherence dishes aren't available, you can use Petri dishes. In this case, you'll have to tap the neurospheres in the morning and afternoon every day for the first 6 days so the neurospheres don't adhere to the bottom of the dish. Add EGF and FGF before placing the dishes in the incubator. These growth factors will need to be replenished every 2 days for the entire length of the culture. On day in vitro 14, fix the neurospheres using formaldehyde to a final concentration of about 4% by adding it directly to the culture and incubating at room temperature for 20 minutes with rocking. Pellet the cells by centrifuging at 300 times G for 5 minutes. Wash the cells twice with PBS and resuspend in a 15 to 20% sucrose solution made up in 10 millimolar phosphate buffer. Leave to cryoprotect at 4 degrees for at least 24 hours prior to sectioning on a cryostat. Remove the cryoprotected neurospheres from the refrigerator and place them in a 60 millimeter dish. For serial sectioning, where a collection of every single section is crucial, I found it best to select neurospheres of good size and shape using a stereo microscope. Using a pipetter, place a neurosphere into a cryomold filled with OCT compound. Mark the spot where the neurosphere is located onto the cryomold. This step will help find the neurosphere during sectioning. Once 5 to 10 spheres have been placed into the mold, rapidly freeze them using CO2. Using the indications on the cryomold, mark where the neurospheres are located directly into the frozen OTC block. Remove the OCT block from the mold and place it onto the cryostat chuck. Fix the mold to the chuck using CO2 and place it in the cryostat. Allow the samples to equilibrate for at least 30 minutes. Once this is done, cut quickly to the marks made on the mold. Then start collecting every section from then on, checking to see where the neurosphere slices begin using a light microscope. Once sectioning is complete, store the slides in a freezer until you're ready to perform immunocytochemistry. For this step, take out the slides containing one of the serially cut neurospheres and include the slides with 5 previous and 5 subsequent sections. This will ensure you image the entire neurosphere. Using a nuclear counterstain will be a must in order to find all the sections of your neurosphere, so keep this in mind as you plan out your immunostaining. Thaw the slides by applying PBS and allowing to warm to room temperature for at least 5 minutes. We use antibody buffer that contains 0.3% Triton X100 for permeabilization and 3% bovine serum albumin for blocking. Dilute your primary antibody appropriately in this buffer and incubate in a human chamber overnight at 4 degrees. Use PBS to float the parafilm off the neurospheres and carefully remove it using forceps. This is the first of 4 5-minute PBS washes. Apply the secondary antibody to slides and incubate them at room temperature for 1 hour. As before, float the parafilm off the slide using PBS and remove it using forceps. Again, this is the first of 4 5-minute PBS washes. In the third wash, we include Hoech's 33258 as a nuclear counterstain, which is diluted to 0.5 micrograms per milliliter in PBS. There is one more 5-minute wash following the Hoech's wash. Once all washes are complete, apply a generous amount of mounting medium to the bottom of the slide and cover with a glass cover slip. Secure the corners with nail polish. Once all slides are cover slipped, secure the rest of the cover slip edges. Image the slide right away or stir them at -20 until you are ready to do so. When imaging, finding the cells on the slide is the hardest part. It will be easy to detect cells belonging to the sphere if you look for them using the nuclear counterstain. Once you find the beginning of the neurosphere, which may comprise only a few cells, image every section from then on, taking shots of the nuclear stain, your protein or proteins of interest, and a phase contrast image. This phase contrast image will be very important in the modeling stages.
Images with residence scale bars of 10 micrometers and 50 micrometers established in OpenLab 3.56 software are imported into Photoshop CS4 using a standard canvas size of 3,300 by 2,550 pixels. All serial section images are saved as TIFF files maintaining the standard canvas size. Open the first and second phase contrast images of the neurosphere serial sections. Increase the transparency of the second section. Under the Image tab, open Image Rotation and choose Arbitrary. Rotate the second section until you are able to identify points of geometric and structural contiguity between images. Repeat for all serial sections, saving each section as a separate layer. Ensure that each image layer corresponding to phase and protein expression are linked so that all rotations for each channel are performed at the same time. Create a plane in Maya that is of the same proportions as the Photoshop file. From the status line of Maya's user interface, change the menu bar from its default animation setting to surfaces. Use a primitive subdivision sphere to create the cell body. With the sphere selected, begin manipulating its vertices by right-clicking the right mouse button on the object and selecting Vertex from the marking menu. Further elaborate the surface of the sphere by changing the display level from the marking menu. From the Subdivision Surfaces tab of the main menu, open the Collapse Hierarchy options box and set the number of levels to 2. Collapse the sphere. Change the menu bar from Surfaces to Polygons. With the sphere selected, open the Sculpt Geometry tool from the Mesh tab of the main menu. Refine the surface of the cell to its final form, employing the Sculpt Geometry toolset from the Tool Settings tab. Repeat this process for each cell of your typology to simulate the variety of progenitor cells present in the neurosphere. In this demonstration, 10 differing progenitor cells will form the basis of the neurosphere. Select the cell typology and freeze transformations. With the cells of your typology established, 2 surface shaders will be designed to represent both cells expressing the protein connexin 29 and cells where the protein is absent. Open the Hypershade window from the Windows Rendering Editors tab of the main menu. Choose a ramp shader from the Surface Materials tab. Open the Attribute Editor and set the shader's color, incandescence, ambient color, bump map, and specular color attributes. Assign a Brownian 3D texture to the ambient color node and a 2D fractal texture to the bump mapping node. Select the resultant shader's input and output connections and export the selected network. Select the cell typology and open the Export Selection options box. Choose the file type Maya ASCII and uncheck the Include These Inputs box. Export Selection. Open the Align Serial Sections file in Adobe Photoshop. Establish a key of pencil strokes to mark the location of each progenitor cell in the serial sections. In this demonstration, a key of 3 strokes, a solid square, a square with a dashed line, and a square with a cross will be used to indicate whether a cell is located in 1, 2, or 3 sections. This key of strokes is further defined by color, red representing cells expressing connexin 29 and white representing cells not expressing connexin 29. Turn the phase layer on and begin marking the center of each progenitor cell with a white pencil stroke. Use a separate layer within the section folder with which to build your maps. Toggle between sections to locate the cell's position, ensuring that cells on more than one section are properly denoted. With the CX29 layer turned on, select the cells that fall in the regions where CX29 is positively expressed. Choose the Fill command from the Edit tab of Photoshop's main menu bar and change the white strokes to red. With the maps completed, save each section as a separate JPEG image in the Source Images folder of the Maya project. To do this, a new Maya project will first have to be created. From the status line of Maya's user interface, change the menu bar from its default animation setting to polygons. Import the planes.ma and scalebar.ma files from the project's scene folder. With the plane selected, assign a Lambert shader by clicking the right mouse button on the object and opening the Assign New Material tab. In the Attribute Editor, click the checkered box to the right of the Materials Color attribute. From the pop-up menu, choose File from the 2D Textures section. Under the File Attributes section in the Attribute Editor, click the file icon to the right of the image name attribute. Choose the first section from the Source Images folder. Switch the camera of the workspace window from the perspective view to the default orthographic top view by opening the Panels tab. Select the Create Polygon tool from the Mesh tab of the main menu and trace the outline of the section. Choose the first map from the Source Images folder and assign it to the newly created polygon plane following the steps established to create the polygon plane. With the object selected, choose UV from the marking menu by clicking the right mouse button over the plane. Select all the plane's UV points and open the UV texture editor from the Windows tab of the main menu. Scale the UVs proportionally to fit the section plane. Repeat this process for each section of the neurosphere, ensuring that the spaces between each section correspond to the height of the scale bar. From the status line of Maya's user interface, change the menu bar from its default animation setting to Dynamics. Leaving only the first section of the neurosphere visible, hide all other sections by changing the display settings from the Display tab of the main menu. With the CV Curve Tools options box open, select one linear from the CV curve settings. Viewing the scene from the top view camera, begin assigning points to the cell maps, creating one curve for the CX29 negative cells and the CX29 positive cells. The resultant curves are flat when viewed from the side view camera. Approximate the thickness of the sections by shifting the points of the curve in the y-axis using the scale bar as a guide. Repeat this process for each section of the neurosphere. Import the cell typology.ma file from the Scenes folder of the project and duplicate the cell typology for both the CX29 negative and CX29 positive progenitors. Open the Hypershade window from the Windows Rendering Editors tab of the main menu. Import the previously built shaders, assigning them to the cells of the typology. From the Outliner window, remove the file name from the beginning of the imported objects. This will become important in later stages of the modeling process. Select the Particles tab from the main menu and create a particle emitter for the first CV curve. Open the Particle Shape 1 tab, and under Emission Attributes, change the max count from -1 to the number of points on your first curve. In the Render Attributes tab, change the particle render type to blobby surface. Click on the current render type box below and change the radius of the particles to 0.01. Attach the particles to the vertices of the curve by first selecting the particles, then shift-selecting the curve. Open the Goal options from within the Particles tab of the main menu and set the goal weight to 1. Click Create. Select the cells from number 1 to 10 in order in the Outliner window and open the Instancer Replacement Options box from the Particles tab of the main menu. In the Instance Objects box, all 10 cells should be listed in order. Choose the correct particle shape name from the drop-down list of the Particle Object to Instance tab. Click Create. By default, only the first cell that was selected will replace the particles along the curve. In order to get all 10 to appear and cycle randomly among the particles, an expression is needed. A second expression will ensure that the cells emit randomly in the same way every time the range slider is repositioned. With the emitter selected, open the Attribute Editor. In the Particle Shape tab, under Add Dynamic Attribute, click on the General box. Under Long Name, write random_index. In Attribute Type, switch from Scalar to Per Particle Array and hit Add. In the Per Particle Array Attributes tab, a random_index box has been added. Clicking the right mouse button over the empty space, select Create Expression from the drop-down box. Type the following text in the expression box. To complete the expression, open the Instancer Geometry Replacement tab in the Attribute Editor, and in the General Options Object Index, select random_index from the drop-down list. Bring the time slider to the first frame and hit play. The cell types are now randomly distributed among the particles. Repeat this process for each section of the neurosphere. Ensure that each new emitter, instancer, and random index is differentiated by name and organized appropriately in the Outliner window. In the Render Using section of the Render Settings window, change the render from Maya Software to Mental Ray. Open the Quality tab, and under Ray Tracing, change the reflection setting to 0. Open the Frame Buffer tab and change the color clip from RAW to Alpha and uncheck Pre-Multiply. Open the Channel Box Layer Editor and change the Layer Editor settings from Display to Render. Select the imported cells of the typology and click the Create New Layer and Assign Selected Objects icon. Rename the layer Ambient Occlusion. Clicking the right mouse button over the newly created layer, select Attributes from the menu. To the right of the Render Layer box, click on the Presets button and select Occlusion from the drop-down list. Select the MIB_AMB_Occlusion1 tab and change the samples from 16 to 256. Reopen the Channel Box Layer Editor and under the Options tab, open the Render All Layers options box. Select Composite and keep layers. With the ambient occlusion layer selected, change it from normal to multiply from the drop-down box just above the layer list. Render 2 passes of the neurosphere, once with the ambient occlusion layer turned on and once with the master layer turned on. Save the images as IFF files in the Projects Images folder. Open both images in Adobe Photoshop. Place the ambient occlusion layer on top of the master layer and set it to multiply. Create a background and flatten the image.
We've just shown you how to culture hippocampal-derived neural progenitor cells as neurospheres, how to serially section neurosphere cultures using a cryostat, and how to perform immunostaining for a protein of interest.
We have also shown you how to model the spatial location of that protein expression in 3 dimensions using a combination of cell culture, registration, and 3-dimensional modeling software. We hope this helps you, and good luck in your experiments.